Welcome to the Metabolic Classroom. One of the reasons I'm frustrated by the modern nutrition obsession with carbohydrates is that there are so many little harmful molecules inherent to these carbohydrates that never get any attention. One of the most relevant, and I hope that after this you're going to consider them somewhat infamous, are lectins. Lectins are one of the more prevalent and problematic, and they have become a significant topic in nutritional science. These little proteins are found in many common carbohydrates, and as you're going to learn, they affect processes like gut function, including permeability, insulin signaling, and inflammation. So, for those seeking to optimize their health, understanding lectins is essential. Modern diets, often rich in things like legumes, grains, and other processed carbohydrates, can expose us to lectins in ways that impact susceptible individuals in particular. Now, the research that we're going to review is going to highlight the effects on permeability and metabolic consequences, all of which really has an impact on our cardiometabolic risk factors and diseases. So, the discussion, the mini lecture, we're going to explore the cellular mechanism behind lectins and their effects on health. And we're going to review a bunch of studies in the process, covering a bunch of models from cells to rodents to humans. So, as we get started, let's understand lectins. Lectins are proteins, as I said, that bind specifically to carbohydrates, such as sugars or polysaccharides, on the surfaces of various cells. Now, they're prevalent in plant foods, which means carbohydrates, most especially legumes like beans or lentils or peanuts, grains like wheat, rice, and barley, and also nightshades, which are tomatoes, potatoes, eggplants, and similar. But they're also found in things like nuts and seeds. Now, in plants, lectins function as a defense mechanism, deterring consumption by other animals or insects by binding to their gastrointestinal lining, causing some gut irritation, and even potentially contributing to a level of toxicity in the body of whoever is eating it. This is reflective of something I've said before, which is that all things that are living want to stay alive. So, by chemical or by claw, something is going to attempt to defend itself. And lectins represent one of the chemical defenders that plants will use. Now, certain lectins, such as phytohemagglutinin in kidney beans or wheat germ agglutinin in whole wheat, and even another one in jack beans called cancavalin A, will vary in structure. And there are others, many others that I haven't even mentioned, but they have a variety of structure and their specificity in what they're binding. But what they have in common will be their interactions in myriad cellular processes that we're going to go through. Now, in our wisdom, we humans have learned that we eat plants and they hurt us. And so it's no surprise that we have developed methods in order to reduce them. So, cooking methods like boiling, pressure cooking, fermenting will significantly reduce lectin content. And in fact, one of the citations for you insiders to access and read is a 2004 study in Journal of Agricultural and Food Chemistry, which demonstrated that pressure cooking can decrease lectin levels in legumes by up to 95%. You can't get rid of them entirely, but these residual lectins will persist, and their ability to have some of the effects that we're going to elaborate are real. And the degree to which various people may experience some response to them is really individual. And so those who are more susceptible to these kinds of things are the ones who, of course, need to be the most careful. But they're going to be the ones who not only feel the gut problems, maybe feel the cardiometabolic problems as well. All of which is just further evidence that perhaps in our revision of nutrition that happened over the past 60 years or so, perhaps we got it wrong when we vilified fat and we inadvertently embraced what is turning out to actually be the villain. Okay, so talking about lectins, let's start in the gut, where so many interactions with food do begin. The intestinal epithelium is the single layer of cells connected by tight junctions. So when you think about the gut, the digestive tract, think about this tube, and the innermost layer, or in other words, what the food and stuff that we are swallowing is going to interact with, is going to be that epithelial layer. It's these cells that are tightly locked together, and indeed, those are called tight junctions. But then the epithelium itself is coated with the carbohydrate-rich glycocalyx. And that is going to be a primary site of interaction with everything we eat, but most especially in the case of this mini lecture, lectins. Lectins bind to the glycocalyx, which then allows them to disrupt gut integrity. A 1999 study published in the journal Gut found that phytohemagglutinin from kidney beans caused intestinal damage in animals, increasing gut permeability. This then means, no surprise, that lectins induced inflammation, disrupted the tight junctions, so making the cells more loosely adhered to one another, opening the gap, making it easier for bacteria to translocate from the gut into the bloodstream. So that's when they're slipping through between the cells, whereas what's coming from the gut is supposed to be going through the cell, because then the cell can act as a gatekeeper in deciding what can come in. Well, when something can slip through, or between the cells rather, now there's no regulation. That's leaky gut. And the leaky gut, of course, can drive systemic inflammation, influencing things like insulin resistance and cardiometabolic disease. In humans, excessive lectin intake, particularly from raw or undercooked legumes, could really exacerbate gut issues, especially in people with things like irritable bowel syndrome, IBS, or various autoimmune conditions, particularly of the gut, like Crohn's disease or ulcerative colitis. A 2017 case series in the Journal of Clinical Gastroenterology reported symptom improvement in IBS patients following simply removal of one thing, namely lectins. Systemic inflammation from gut disruption elevates cytokines like TNF alpha and interleukin 6, all of which can be easily measured in human plasma and is. In fact, another one would be C-reactive protein, which is measured nowadays in practically every blood test you're going to get. And the more you have inflammation up, these markers elevated in the plasma, the more you have the consequences like insulin resistance. And of course, properly preparing the food in order to try to mitigate the amount of lectins you're consuming is going to be essential. But again, you cannot remove all of them. Now, I mentioned insulin and insulin resistance a little, and in fact, lectins go a little more direct. They are particularly relevant because lectins can literally interact with insulin receptors. So these are the specific receptors, the doorways that are designed for insulin to come and knock on them to tell the cell to do something. Well, wouldn't you know it, wheat germ agglutinin, which I'll just abbreviate as WGA, found in whole wheat is a notable example. A 1980 study in Proceedings of the National Academy of Sciences demonstrated that WGA binds insulin receptors in fat cells. Now, of course, it would be happening at any insulin receptor, but at low concentrations, WGA mimics insulin, stimulating things like glucose transport and inhibiting lipolysis and promoting lipogenesis. So just overall promoting the growth of the fat cell. At higher concentrations, it actually starts to antagonize insulin, reducing the binding affinity and inhibiting glucose uptake, which can then contribute to problems like hyperglycemia. So the particular study, which was done in fat cells directly, showed that WGA's insulin mimicking effects weren't as strong as insulin's. So it required a higher concentration to achieve these same results, but it nevertheless is evidence of this interaction, this cross interaction of this seemingly inert molecule found in these carbohydrates that are not only think about the whole thing here, the whole story, where you're eating like wheat, for example, whole wheat, which is going to elicit a considerable increase in your glucose, which in turn is going to elicit a considerable increase in insulin. So not only do you have the inherent normal insulin load, which as it persists can contribute to insulin resistance. But now you also are consuming these other little molecules that are mimicking insulin's effects. So you're amplifying an insulin response. And remember, the more you have the insulin receptors getting stimulated, the more they're going to become desensitized to the signal. Now, talking about insulin makes it easier and easy to transition into other cardiometabolic problems just because of insulin's influence on cardiometabolic health through so many mechanisms. Well, once again, lectins are very relevant here. Let's start just again by revisiting a little more evidence on adipose tissue in the context of obesity. Inflammation in adipose tissue is a hallmark of insulin resistance. I've said this before, that much of chronic slow insulin resistance begins in the fat cell or adipocyte. A 2016 study in Journal of Nutrition found that animals fed high lectin diets, like that including legumes, exhibited increased fat mass and adipose tissue inflammation compared to controls. Importantly, this is even after controlling for things like calories. Lectins triggered immune activation, resulting in an increase in cytokines like C-reactive protein. And again, remember, the more inflammation you have, the more insulin resistance is going to follow. And so all of this leads to a disrupted metabolism of fat within the fat cell. And as they found, it promoted fat accumulation. The study also observed macrophage infiltration into adipose tissue, which is a strong feature of obesity-related insulin resistance. So let me just paint that picture. Within the local neighborhood of fat tissue, we would have the invasion of macrophages. So basically, a neighborhood consisting of just fat cells now has these angry, very pro-inflammatory macrophages moving in. And you can literally see this happening when you are looking at a staining or an image of the fat cells, of the fat tissue. You can see a bunch of macrophages. All right, now let's move on to cardiovascular effects. Lectins can also affect cardiovascular health. A 2002 study in toxicology showed WGA, that's the wheat germ agglutinin, increased platelet aggregation in vitro. Now, this was a cell culture type study, but when the lectins went up, the platelets were binding together more. And of course, this then raises the risk of things like thrombosis or a blood clot. A 2013 study found that peanut lectins directly activated inflammatory pathways under the control of a master regulator of inflammation called NF-kappa B. And this specifically happened in endothelial cells. The endothelial cells are the cells that line the blood vessels. And this, in turn, upregulates these things called adhesion molecules, like ICAM1, for example. So it makes it more likely for things like macrophages to get stuck along the sides of the blood vessels. And that is a pivotal event thought to contribute to atherosclerosis, so plaque formation. And even with the blood vessel, like the endothelial cells, insulin is relevant. Endothelial dysfunction reduces insulin's vasodilatory effects. So insulin normally, when it comes through a blood vessel, will tell the endothelial cells to change the diameter of the blood vessel. And the more you are widening blood vessels, the more blood pressure is going to be controlled. Well, lectins are disrupting that. So we have this chronic sort of vascular level inflammation contributing to a lack of vasodilation, contributing potentially to the direct formation of the plaque by making it easier for macrophages to stick to the walls of the blood vessel and then get into the blood vessel wall, which becomes the core of the plaque. Okay, so that's a little bit on adipose tissue with the direct evidence of it contributing to obesity, even when calories were controlled. The effect on the blood vessels. Let's now talk a little bit about the liver. Lectins may harm the liver by promoting inflammation and insulin resistance. Now, to understand the effect on the liver, we actually have to have a bit of an indirect effect here too, which is leaky gut. A leaky gut allows bacterial endotoxins like LPS, lipopolysaccharide, to reach the liver. Remember, when you look at blood flow through the visceral space, when blood flows through the liver, rather through the gut, it then goes to the liver. So the liver is really on the front lines of what's coming in from the blood is going to get to the liver. So if we have a lot of endotoxins like LPS moving from the gut because of leaky gut, the liver is the one who has to really deal with it. So it's no surprise that if there is a leaky gut, the endotoxins will reach the liver, which in turn activates the macrophages in the liver, and those are specifically called Kupffer cells. And so these liver macrophages get really primed for inflammation and start releasing a bunch of cytokines like TNF alpha and C-reactive protein. But the hepatic or liver inflammation, in turn, impairs insulin signaling, creating hepatic or liver insulin resistance. This means more gluconeogenesis, less glycogenolysis, and more lipid synthesis. So the liver basically is getting loaded with glucose and loaded with fat. So no surprise that there's an increase in non-alcoholic fatty liver disease. In fact, a 2018 study in Journal of Nutritional Biochemistry found high lectin diets increased liver fat and insulin resistance, linked, no surprise, to elevated inflammatory markers. But it doesn't stop there. Lectins, like the wheat germ agglutinin in WGA, can also induce oxidative stress in hepatocytes, liver cells, potentially causing mitochondrial dysfunction and contributing to steatosis or fatty liver. So in individuals with autoimmune predispositions, once again, lectins' molecular effects could trigger some specific liver issues. Now, one final big thing I wanted to mention about lectins is their role in aggravating immune function. Lectins can stimulate immune responses. A 2015 paper in an autoimmune journal called Autoimmunity Reviews proposed that lectins act as, I like this word, molecular mimics. So, in fact, molecular mimicry is an area of research exploring how some of these unknown or previously unknown components or molecules of various carbohydrates can trigger cross-reactive antibodies that worsen autoimmune conditions. So this is basically just saying that the lectins are priming the immune system to start to recognize self or our own cells as foreign. A paper in 2019 found lentil lectins induced interleukin 6 production in human immune cells, which not only disrupted insulin signaling, but again, is just contributing to this overall pro-inflammatory, pro-autoimmune condition. Lectin's structural similarity, that's reflective of the molecular mimicry, to our own proteins can confuse the immune system. And like I mentioned the WGA, but also phytohemagglutinin. They can bind to the gut epithelial cells or immune cell receptors, and they can mimic some of our own proteins, which can prompt the production of auto antibodies or self-destructing antibodies. One of the other papers I have here highlighted how lectins can initiate or amplify autoimmune diseases like rheumatoid arthritis or lupus by stimulating T cells. T cells are one of the critical white blood cells involved in immune responses. So this immune dysregulation may extend to not only systemic inflammation, where we can see elevations in cytokines in the blood, like C-reactive protein or TNF alpha or the interleukins, but also contributing to liver dysfunction, adipose dysfunction, etc. But of course, the autoimmune link also ties to cardiometabolic health. Chronic inflammation from autoimmune activation increases cardiovascular risk, as seen in conditions like lupus, where patients face higher rates of atherosclerosis. A 2017 study in clinical rheumatology noted that autoimmune-driven inflammation correlates with insulin resistance and endothelial dysfunction, both exacerbated by dietary factors like lectins in susceptible individuals. So once again, while we do have inherent mechanisms or cultural mechanisms to try to reduce these lectins, like just how we prepare foods, you never get rid of all of them. And the risk in those people who just tend to be a little more susceptible is very real. Okay, so let's get to some practical considerations. And I've been touching on this a little bit, but a lot of lectin's effects on health, including all the things I've outlined, depend on the preparation, the dose of how much you're eating, and individual susceptibility. Cooking methods like pressure cooking or boiling can significantly reduce a lot of these lectins, which is going to minimize the exposure and minimize the risk. But people with IBS or autoimmune conditions or insulin resistance or plaque problems like atherosclerosis or high blood pressure may benefit from deliberately limiting these high lectin foods, like controlling raw grains or legumes. In fact, that's an important point, that while it's exceptionally uncommon for someone to eat raw legumes, it's not uncommon at all for people to eat more raw grains, or they haven't gone through this process of, say, high pressure cooking. Systemic insulin resistance, inflammation, and cardiometabolic problems are influenced by a host of factors, but the drum that I beat so consistently is control carbs. Now, much of what I've discussed over the years with control carbs has been focused on the insulin effect of those refined starches and sugars, but we cannot ignore some of these more hidden aspects like lectins. Now, one thing you can do then is monitor biomarkers like fasting glucose and insulin, of course, but also high sensitivity C-reactive protein. That's going to be a pretty good one that may let you know, may give you some insight into whether you're eating something that's actually making your body have some kind of immune response. Now, a lot of the studies I mentioned are done in animals or cell culture with a little bit of human data, so we need more human studies to really quantify lectin's role in cardiometabolic health, but it is obvious in the limited data we do have and the abundant anecdotes that limiting lectins is going to be a generally helpful intervention.